Hello, welcome to this presentation. My name is Karina Cuanolo Contreras and I am the Scientific Marketing Manager of Savoy. At Savoy, we have more than 30 years of experience in the processing and manufacturing of membranes and plastic for life science and cell culture applications. My colleague Vivian Monteban and me are completely delighted to have the opportunity to present in the ACTC conference, and we would like to thank the organizing committee for this invitation. Our goal today through this presentation is to create awareness regarding the challenges that are faced toward new in vitro animal-free methods and how can we use these challenges as opportunities to accelerate the in vivo to in vitro transition. We would like as well to open up a forum for discussion where we can exchange our ideas and work together as a team to make these methods the new standard. To start this presentation, I would like to provide some data regarding animal research. It has been estimated that every year, more than 9 million animals are used for research purposes in the European Union. 49% of them account for basic research with the goal to help advance scientific knowledge. 23% go for translational research, aiming to understand diseases and developing new medicines. Other 23% are used for the regulatory assessment for potential risk of drugs, chemicals, fertilizers, and food additives. The rest will go for education and training, as well as the creation and maintenance of genetically altered cell lines. The most common animals used in research and testing are mice, accounting for 61%, followed by fish, rats, and birds. Whereas all these in vivo methods have played a key role in the medical advances in the last century and have been the gold standard for preclinical and toxicology research, more and more researchers are increasingly transitioning to in vitro testing. For example, in the European Union, the use of animals for research and testing has been reduced 22% in a period of 10 years, supporting the efforts toward the 3Rs principle of replacement, reduction, and refinement by minimizing or replacing the number of animals used for scientific procedures and reducing their potential distress. Now, several key driving factors are involved in this growing transition. Generally speaking, in vivo methods are costly and can be very time-consuming, especially when considering long-term exposure studies. In addition, they are technically challenging and impose many ethical concerns. Therefore, their high throughput potential is rather limited, and the control of the chemical and physical environment is low, making it difficult to draw accurate conclusions. Just to give you an example, it takes about 4 to 5 years to get results based on animal testing for cancer bioassays. That will cost around 1 million per chemical and animal species tested. And this is not even considering the high false positive rate, where 53% of anything tested opposed to 5 to 10% true positives. Apart from these aspects, one key factor that questions the pertinence of the use of in vivo methods is their relevance to human health. The poor predictability of animal experiments is one of the major challenges in the field. The use of non-human animal species and different strains with a variety of metabolic pathways leads, of course, to variation in efficacy and toxicity, making this cross-species extrapolation a very important issue. Uncertainty in safety and efficacy assessment come from reproducibility issues, mainly due to the intrinsic biological variability and the inadequate statistical power that comes together with the use of small groups. In addition, animal models often do not accurately predict adverse reactions or recapitulate important aspects of disease in humans. For example, in the field of neurodegeneration, Alzheimer's disease transgenic mouse models fail to fully recapitulate the very basic physiopathological aspects of the disease, such as synaptic alteration and neuronal death. On the other hand, in vitro methods allow the use of human cells, eliminating the issue of cross-species variability, and can help reveal mechanistic information as to how a substance may exert biological effects, providing human relevant insights to make more accurate predictions. In addition, their scalability and high throughput potential increase reproducibility and statistical power. The development and use of advanced in vitro systems has increased their physiological relevance and closely mimic the in vivo microenvironment. One example is the use of brain organoids generated from human-induced pluripotent stem cells. These models mimic the complexity of the brain architecture and organization. They offer a great alternative to animal models for screening of drugs, with the added advantage that all the components are of human origins. Another very important example is the use of human barrier tissue models that grow on cell culture inserts, such as lung and intestinal models, blood-brain barrier, and skin. They are widely used for toxicological research and ADME testing due to their human relevance. However, despite the amazing advances in the in vitro field, there are still many challenges. Among them, some in vitro methods may have the potential limitation to replicate the conditions of cells in tissues, organs, and systems, especially static methods based on monocultures. The legislation and regulatory acceptance is another big challenge in the field, as historically animal testing has been the gold standard. However, currently there is an increased focus to better understand how exogenous substances exert their effect at the cellular level. Some in vitro methods can be technically demanding and expensive, particularly in terms of initial setup, training, and equipment cost. However, as technology advances, it is expected that these methods will become more and more accessible with time. And finally, while many in vitro alternatives do not involve animals directly, animal-derived products such as fetal bovine serum may still be required. But the important topic here is, how can we use those challenges as opportunities? Through the development of advanced in vitro technologies, it is possible to increase the physiological relevance of the models, and currently there are many groups working towards this goal. For example, in the field of microfluidics, tissue barriers, and organoids. Legislation changes have been a driving force toward non-animal research, with the advances of the in vitro methodologies made in the cosmetic sector and used in regulatory safety testing. It is anticipated that confidence will be given to other sectors to move away from the traditional in vivo models. For example, in 2013, the European Union banned the marketing of cosmetics containing ingredients tested on animals, and other regions have since followed. Another example, in 2017, the European Union regulation for the registration, evaluation, authorization, and restriction of chemicals was amended to make in vitro testing the default for skin, eye irritation, and skin sensitization. The fetal calf serum-free database has been also set up to provide information on animal-free media for cell culture. In addition, there are many funding opportunities towards the 3R. And last but not least, we would like to highlight the importance of the inclusion of manufacturers in the process of developing new animal-free methods. By partnering, we could progress to achieve the goal to develop more powerful and reliable methods that mimic the complexity of higher organisms. This is, of course, not trivial and requires the cooperation and exchange of knowledge between scientists and industry. In this regard, Savoy is an original manufacturer guaranteeing 100% parameter consistency and actively seeking ways to put their expertise in the production of microporous membranes and plastics to use in advanced cell culture systems that are relevant to human and animal health. We would like to conclude this presentation with the following remarks. It is fundamental to acknowledge the advantages and limitations of in vivo and in vitro methods, including both ethical and regulatory considerations. By applying the 3Rs towards the in vitro transition, in vivo testing can be minimized without affecting the rigor of scientific research. Savoy is actively working to accelerate this transition. For example, our trackage membranes can be found in state-of-the-art on-a-chip systems, and in 2020 we launched our home brand cellQART, cell culture inserts. We are making a new standard. Please join us. We have the expertise, we are quality driven, and we can really make an impact helping you to reach your goals. We would like to thank you for your attention. Please share your thoughts and let's make this new approach methods the new standard. Thank you.